Hello, welcome to this video. This video is possibly the last video of the series that we have started, and in this video we'll be touching and looking at how to visualize our complexes and then determine the different interactions that we have obtained from our docking, all right, between our protein and our ligand. So that's what we'll be looking at two in this video. So all right, so um let's see what we get here. So we actually have we started and we stopped at where we got our binding energies, right? Yeah, so the binding affinity, and then okay, let's open it at the results of the binding the docking, right? Yeah, so this is what we got, and this is our ligands here, the results that we got right here, right? Okay, let's say, yeah, so the binding energy. So this is our result. So let's work with this one, the first one, with the binding energy of minus 6.9. Sorry. So we will be working with that. All right. So what we want to do now is so we have our prepared proteins here, right here. Yeah, we will be needing it very soon. So we want to open macromolecules here. Yes, right. So this right here is what we are looking at too. So what we want to do is to open this file, pdbqt file, in WordPad format. All right. So open with WordPad. So yeah, we've opened this, and then we will open. Okay, let's open this. Our prepared. All right. So this is it. Oh, this is our prepared protein. Okay. Let's see. Yeah. So at the end we have terminal there. All right. So what we want to do now, we want to check. So in this file, you will see the different models, all right, that we obtained even from the docking. You will see model one here, model one here. All right. So yeah, model one. All right. So and then um, so just to say that there are different models. Okay, this is model two, model 3, etc. And you will notice that for every and each model, the binding affinity might be different. Here, for example, we have minus 6.7. So we want to take the model that has given us the binding affinity of minus 6.9. All right. So it's for the same ligand, but remember when we were doing the virtual screening, the docking in Barracks, we said that there are different, yeah, according to the models, you can have a similar binding affinity for the same ligand, but not exactly the same. All right. Yeah. So that is what we are observing right here, right here, minus 6.7, and the model 2 is also giving us minus 6.7. So we are looking at to the model that gave us minus 6.9, and this is it right here. So model 1. So from this model 1, we are interested in everything hetat heteroatoms. Everything heteroatoms we are interested in. Okay. So what we want to do is, so from here we want to copy it. heteroatom till we find what? Model 2. So model 1 stops here. So let's copy it. We copy it, Control C. We come to our prepared protein, okay? At the end, where we have there, TER, okay? So we are not interested in this TER, okay? So we have deleted the TER. We now paste what we have copied from yeah, from our complex, the output, right here. Okay, so now we don't want anything end road, all those things, anything that is not heteroatom, like I mean HETATM, anything that is not that, we have to delete them. End root, we delete it. Branch, yeah, we delete it. End branch, delete. Branch 8, yeah, delete. End branch, toss stuff, yeah, deleted. So now we have what we needed. So this is how we form our complex, all right? Yeah, to be visualized. So now we want to save. To save in another format, click no. You are about to save the document in so yeah, Control S. Test only format. I say no, and then I want to save it as PDB. PDB, and what am I looking for? All right, so network farm discuss this right here. All right, so yeah, so I added the pdb.pdb so that it can be saved in pdb format. It says already exists. Okay, no problem. But what I want to do, okay, let's name it complex. All right, so let me rename rename it. So, and the ligand, let me say ligand, and then complex. All right. Let's check whether it is already there. So right here. It's not here. What happened here? Let's see. I say no. Ligand complex dot daily b. I say yes. Let's see. Alright, so is there now? So this is it right here. Okay, so this is our complex. Let's open it and let's open it in Discovery Studio. Yeah, so this is our complex. Alright, this is our complex. Alright, so this is the protein. Let me open it from here so that you can see the protein. Say protein group, a yellow there. Alright, then the ligand group. This is our ligand in yellow right now. So this is our complex. Okay. All right. So we have obtained our complex here in Discovery Studio, right? So now we want to see the different interactions. So let's click on ligand interaction. Let's see. Okay, let's do this first. Show to the. Okay, so immediately you click on show to the diagram. So it will show you the 2D diagram, the different like as you can see it here at your right side, okay, of the screen. You have the different interactions. So this is the ligand right here, okay, as you can see it. And then the circled and colored, yeah, the colored circle, green, purple, yeah, that you are seeing are different amino acids of our target, the protein, all right. And the dashed lines are the different interactions between the ligands and then the uh, the amino acids of the protein. So you can see right here down here in the different interactions, the colors with the indicated type of interactions. So it's saying that these colors, this green one, light green, is Van der Waals interactions. And should I see the, should I say the deep, is it deep green now or light green? Oh, yeah, but I guess you understand the colors that I'm talking about, which is showing the conventional hydrogen bond. All right, conventional hydrogen bond. Uh, the purple one, okay, um, pi sigma bond, and then the this is pink, kind of purple light, yeah, alkyl and pi alkyl interactions. So here you have your interactions in a 2D diagram, and you have the different interaction, the different types of interactions, and the interactions with, and they have different amino acids within um, with which the ligands are interacting with. All right, yeah. So this is it. And if you want to how do you call it now, to save this particular image, you just come on file, all right, save as, and make sure you have selected this particular file, all right, because if you select this, that means you are working with this. But you select this and you are sure that by the moment you say save as, when we click on save as, we can save it. This is not it. You can see that the files that come says image files, all right. So you know that you are rightly working with oh, net. Okay. All right, so let's save the complex here. So we just save it here. Okay, it says save image. So you can change the criteria, the width, height, pixels. You can save, you can change them. Then you just say okay if that's what you want, and it is saved. Let's check. So this is it here, right? Okay, so this is it. All right, so we have it. Now, I want to say something. Let's close this. Suppose we didn't ask the Discovery Studio to show us 2D diagram. and or we are not interested in the 2D diagram. All right, so we say ligand interaction. Let's click on ligand interactions. So this is after clicking on it, this is what is showing us. So this is our ligands, and then the different interactions. All right, yeah. So if you notice, we have this will not change the interactions. All right, whether you have it in 3D or 2D, it will not change your interactions. You will notice that we still have the same kind of interactions, all right, as we had them in the 2D. Yeah, so this is what we are getting. Okay, all right. So so he's saying display receptor surfaces. If you click on HG, yeah, aromatic, or you want the charge to be displayed, surface area, um, I mean solvent accessibility, hydrophobic, depending on what you want to put out there. All right, yeah. But this for me is usually not the um not the not an important thing. But then uh it says show types. Yeah. So all right. So we were talking about the interactions options, show types. So if you click on show types, it will show you, it will write down the interactions. As we saw it in the 2D diagram, it's showing that these green ones are usually the conventional H bonds, and usually we are interested in the conventional H bonds. Yeah, I'm not saying the other interactions are important as well, and is good to show them. Yeah, but generally, drug discovery, they are looking out to the H-bond interactions, all right? So you can say show them or not, okay? Yeah, so show types, conventional, and then if we turn this, we will see pi sigma, pi alkyl, all right, I alkyl, yeah. Now, what we want to do, or if you want to show the distances, all right, of the interactions between um, your ligand and the proteins. Now, here we can see that we are not actually, we are not seeing the how do you call it? The amino acids. So how do we do that? So right click, all right, there, and you say, um, okay, labels, and you say add labels, all right. So atoms, object, we want to change the labels, all right. So we want to see the amino acids, okay. So you can change color here, you can change the font here, and the size, all right, yeah. So you click on apply. Immediately you apply, you will see that they are shown, all right, the amino acids, all right, they are shown, all right. So you say okay, and you can see the different amino acids, all right. Valine, lysine, leucine, glycine, okay, alanine, yeah, etc., etc. If this is too big for you, not containing in the same in this view, in this framework, all right. So you can just come here, you say mouse mode, you say zoom. So the our mouse is now in zoom, okay. So you just double click maintain, and by the moment, yeah, so by the moment you manipulate your cursor, it will zoom it for you, zoom in, zoom out as your as you want it, all right. And yeah, so this is it. Okay, so let's try to save this image, right? Let me just close this one. Let's try to save. Okay, let me just say I don't want to show distance, I don't want to show type. Okay, so this is what we get. Okay, let's say we want to save it. File, save as. We want to save as image file. Okay, so let me say dot two, or let me just say two. Save. I say okay. Let's see what it looks like. Okay, so this is what it looks like. Okay. Mm, script visualization, and it says publication qualities. All right. So you click here. Okay. All right. So this is just for if you are publishing for quality, the quality of your image. All right. So just for the quality of your image, just for the quality of your image. So script visualization, publication quality. Yeah, and then you can save. All right, so that's it. Okay, so this is what it looks like in 3D. Yeah, all right. Um, yeah, so that's it. That's how you determine, you visualize your protein ligand complex, and then you determine the different interactions, all right, between your ligand and your protein. What type of interactions, you will know what type of interaction, you will know which amino acids on your protein or target your ligand is interacting at. Yeah, so yes. All right, so this is it for the protein ligand interaction visualization in Discovery Studio. All right.